members network of the Metabolomics Society. Today, me and Mitzi Mogla from the University of Johannesburg will be hosting this live webinar. Don't forget, if you need any more information about the society, you can find our page on Facebook as well as our Twitter account, or you can email us using our email address, which you can see at the end of this slide.
Okay, for this webinar series, we also have a webinar lined up for the 30th of March, which will be presented by Professor Caroline Johnson and Mrs. Anna Rosen-Volmer. And their topics for Caroline, the topics will be metabolomic subphenotyping of colonic cancer prognosis. And for Anna, it will be normalizing untargeted periconceptual urinary metabolomic state. Please make sure that you register for this upcoming webinar, and you do register, and also please attend. In this webinar, the registration will be shared on the different platform that we use to advertise the webinar. And then for today's webinar, we have Dr. María Eugenia Monge, and today she will be presenting, her topic is the TidyMS, a tool for pre-processing LC-MS data in untargeted workflows. And we also have Ms. Nicolás, who is going to be presenting on improving data quality in metabolomics workflow, a clear cell renal cell carcinoma case study. So our first speaker will be Dr. María, and Dr. María is an independent, so I'm going to just read her bio and also what she'll be presenting on. So Dr. María is an independent researcher from the CONICET and the National Science and Technical Research Council in Argentina. In 2006, she obtained her PhD from the University of Buenos Aires. Between 2007 and 2014, she held postdoctoral position in Italy, France, and the USA. In 2014, she was recruited by the CONICET to set up a new laboratory at the Centro de Investigaciones and Bionanosciences, and where she leads the bioanalytical mass spectrometry research group and the MS facility. Her research group applies MS mass spectrometry for biomarker discovery and early disease detection, with a special emphasis on untargeted metabolomics. And Dr. Maria's ongoing project involves the oncometabolomics field, mainly focusing on the study of genitourinary urine tumor, as well as the investigation of danger fever treatment and progression using a combined pharmacometabolomics and biomarker discovery approach. She is a co-author of more than 45 peer-reviewed research publications, and she has coordinated metabolomics courses for South American studies and has participated in promoting awareness of metabolomics with the hope of strengthening the Latin American community. She is a founding member of the Latin American Metabolomic Profiling Society, and since 2019, she has been a member of the Metabolomics Quality Assurance and Quality Control Consortium, and a member of the Metabolomics Society, where she serves as a member of the Metabolomics Society Membership Committee. She has also served as a guest editor for a Journal of Metabolomics, and she is in the editorial board for the member of the Gigabytes Journal. So just to give you a brief introduction, in this webinar, Dr. María will be presenting how one of the challenges in untargeted metabolomics workflow in pre-processing data in a reproducible and a robust way. In liquid chromatography mass spectrometry data, curation involves removing biologically non-relevant features such as retention time, mass pairs, and retaining only those analytically robust enough to allow subsequent data analysis and interpretation. In this presentation, María will share the strategies that they use for quality control purposes in LC-MS-based untargeted studies, and that interrogate samples from in vivo model and serum sample, as well as ambient mass spectrometry-based approaches. So without wasting any time, I'm going to allow Dr. María to present her work to us. And over to you, Dr. María.
Hello, does it look properly? Oops.
Yes, everything looks fine.
Perfect. Well, hello, good morning, good afternoon. Many thanks, Cici, for your kind introduction and complete introduction. And many thanks to Evelina and Michelle Reed also for inviting us to participate in the EMN webinar series of the Metabolomics Society. It's a great honor and pleasure to be here today and to be able to present some of the results that we have obtained so far in our research group, which is located in the Centro de Investigaciones en Bio-Nanociencias, which stands for the Center for Bio-Nanoscience Research, which is actually a national research center from our National Research Council, CONICET, and it's located in Buenos Aires, Argentina. This is a picture of our lab and a picture of our research institute, which is located in the Scientific and Technological Pole of Buenos Aires. So the presentation I prepared for today, this is the outline that involves describing a little bit about the context that led us to the development of TidyMS. But before getting into TidyMS, I wanted to share with you which were the implemented strategies and the quality control practices that we have conducted so far in our research group. And I will illustrate with briefly describing some of the studies that we have conducted so far. And then I'm going to describe the goals and the motivation that led us to develop TidyMS, the capabilities of the tool. And I'm going to illustrate these capabilities with two different applications. One focused on system suitability testing for an LC-MS platform, and the other one focused on signal drift evaluation and a data curation pipeline. I will end with some conclusions about TidyMS, and then before finishing the presentation and based on the suggestions from Michelle Reed, I'm going to introduce you and share some news about the Latin American Metabolic Profiling Society. So to begin with the context, in the last years, there has been increasing awareness in the international metabolomics community about the need for implementing quality assurance and quality control processes to ensure data quality and reproducibility. And this was evidenced by the establishment of the Metabolomics Quality Assurance and Quality Control Consortium, MQAC. In particular, in our research group, we follow several guidelines that were provided by the research groups of Ian Wilson, David Brohurst, Rick Dunn, and Coral Barbas, among others, and all their collaborators. And so in our lab, the goals involve developing mass spectrometry-based metabolomics and lipidomics workflows by using biomarker discovery strategies in order to obtain or find metabolic signatures or fingerprints that would allow us to investigate different health-related questions or environment-related applications. So most of our research projects, as Cici mentioned in the introduction, are focused on health-related applications. In particular, we aim at finding these metabolic signatures or fingerprints that would allow us discovering potential biomarkers for disease diagnosis and prognosis, and for the improvement of the understanding of disease pathophysiology. And with these two pillars, we have conducted several studies focused on the investigation of genitourinary cancers. We also have a project currently starting on dengue disease, and several collaborations focused on the improvement of understanding of cancer metabolism and molecular neurobiology. So far, in the research group, we have interrogated human biological samples from biobanking systems and hospitals, as well as samples from in vitro models and animal models. As I mentioned before, most of the studies we have conducted so far are focused on improving diagnosis and understanding the disease pathophysiology of genitourinary cancers, in particular in kidney cancer and prostate cancer. So a couple of years ago, we revised the literature with my group members, and we summarized in a review article after reading all the literature, focused on mass spectrometry-based metabolomic studies that were aiming at improving the genitourinary cancer diagnosis. We summarize here in these two maps, these two figures, the countries in which research groups have put effort into finding potential biomarkers for disease diagnosis. This corresponds to the scenario until March 2019. And through reading all the literature, we identified several needs. Among them, I have listed here some of them, which includes the need of revising the suggested biomarker candidates among the different laboratories, the need of replication and validation studies in larger and different cohorts, the need of improving the description of quality assurance and quality control practices in order to be able to reproduce and validate results from other labs, the need of reporting the data preprocessing and processing steps, the reporting of confidence levels for identifying metabolites, in particular in the case of lipids, following the suggested rules for annotation, and the need of sharing the data in public repositories. So since most of the studies that we have conducted so far were focused on kidney cancer, I wanted to briefly introduce you about this disease, and then I'm going to briefly summarize and show you results from the studies that we have conducted so far. So kidney cancer is a disease of dysregulated cellular metabolism. It's the 15th cause of cancer-related death worldwide. Approximately 90% of renal tumors are renal cell carcinoma, and more than 20% of patients exhibit locally advanced or metastatic disease at the time of diagnosis. It's inherently resistant to chemotherapy and radiotherapy, and it consists of several histological subtypes, among them being clear cell renal cell carcinoma, the most prevalent, which is also considered to be a glycolytic and lipogenic tumor. And it's classified into stages 1, 2, 3, and 4. Unfortunately, more than 50% of renal cell carcinoma patients are incidentally diagnosed at the clinical setting. The diagnosis is currently based on clinical examination, ultrasound screening, CTS, and histological information. It's a disease that is potentially curable by surgery, but only at early stages. So with this scenario in mind, with this context in mind, we implemented in the lab different strategies that involved analyzing samples from in vitro models as well as from human serum samples using different, totally different strategies such as a metabolic footprinting study, a metabolic fingerprinting study, and a lipidomics machine learning study. So in all of them, we aim at following quality assurance and quality control practices and reporting them in the publications. So what I'm going to do now is briefly tell you some hints about each of these publications and which were the quality control practices that we have followed so far. So the metabolic footprinting study of a clear cell renal cell carcinoma in vitro model was the first study that we designed in the lab. At the moment, the group was mainly composed of Dr. Elena Not, Dr. Malena Mansi, and Nicolás Zabalegui, who will talk afterwards, besides other students. And they were, together with me, committed to design a study in order to verify the hypothesis that we had at the moment, that was that conditioned media would be a good proxy, a good serum proxy for studying CCRCC. And so with that goal, we designed a metabolic footprinting study to analyze conditioned media samples that were collected from a control cell line and 2 CCRCC tumor cell lines. And we were wondering if the secretome, that is the metabolites that are released by the cells to the external media, would be a good approximation to find potential biomarkers of disease and a good approximation of the serum samples by comparing and contrasting this secretome with a small pilot cohort of serum samples collected from healthy individuals and stage 4 CCRCC patients. So in this study, we found discriminant compounds, a panel of discriminant compounds from conditioned media samples that allowed us to differentiate stage 4 CCRCC patients from healthy individuals. In particular, in this first study, we analyzed different quality control samples, including a system suitability blank, a system suitability sample, process blank, and culture media blanks. We also analyzed intra-study QC samples that were prepared by pooling equal volume molecules of subsets of the study samples that were also used for conditioning the LC-MS platform. We used commercial serum as an inter- and intra-lab QC and long-term reference samples. The samples were analyzed in a randomized way, and we performed several manual data curation steps such as blank correction, filtering signals based on chromatographic peak shape, signal intensity, isotopic pattern, and prevalence in classes. We analyzed the data with the principal component analysis to evaluate if we were having potential outliers and if the pool quality control samples were clustering tightly in the score plot. And we found important the need of just comparing extracting the feature matrix by just comparing cell lines that were cultured with the same culture media, since the culture media can influence the exometabolome. And we also, in this particular study, we found very important the fact of performing spiking experiments to address retention time shift associated to the concentration factor that we use to prepare the conditioned media samples, as well as to identify different types of ionic species that were detected based on this concentration factor that we use in the form of sodium chloride clusters. And in particular, another point that we found important in this study was to have an experimental design that would allow us to account for biological variance, even if the in vitro models are control systems. We aimed at generating samples starting from 2 cryovials for each cell line and generating 72 samples. Out of them, 6 were used for developing the analytical method and 66 for the analysis of the metabolic footprinting study. And as well, the data from this study and from the other studies were deposited to the metabolites public repository with this dataset identifier. The second study that was conducted in the lab, more or less in parallel, we started with Dr. Elena Knott, followed by Dr. Juan Manuel Gurevich, and then the study was led by Manuela Martinevsky. The 3 of them were postdocs in my group, and Manuela is now a research assistant in the group with my supervision. And the goal was to improve the understanding of CCRCC pathophysiology by designing a discovery-based metabolic fingerprinting study in which we analyzed the endometabolome of cell lines from in vitro models of human kidney cancer. And we wanted to investigate the effect of RSUME, which is a protein that is known to be expressed in patients with clear cell renal cell carcinoma. And we wanted to evaluate the effect of this protein in 786O cells that had or not the von Hippel-Lindau gene, since in CCRCC, between 50 and 80% of patients have mutated this gene. And so by means of different statistical techniques, we found discriminant metabolites that allowed us to understand and unveil the role of this protein into these systems. And the study was complemented with bioinformatic analysis and a biological analysis. In particular, for this particular study, we conducted different similar QC practices as in the previous study. We also analyzed system suitability blank, system suitability sample, a process blank, intra-study QC samples. In this case, they were pools of equal volume molecules of extracts of all study samples that were also used for conditioning the LC-MS system. And we applied a 100% detection rate filter in pool QC samples that allowed us subsequently to correct for the locally estimated scatter plot smoothing signal correction method applied by using the pool QC samples. We also conducted blank correction and prevalence filters and retained features under a threshold of relative standard deviation in pool QC samples. We inspected the data by means of PCA, and we also applied the different manual curation steps that I mentioned before. And in particular, in this study, we also found an important point related to the experimental design that was to analyze a large number also of samples from in vitro models that allowed us to evaluate the confounding effect of the culture media in the endometabolome profile. Again, the data was deposited to the Metabolomics public repository and can be found with this dataset identifier. The third study that was conducted to investigate clear cell renal cell carcinoma comprised lipidomics and machine learning strategy and was conducted mainly led by Dr. Malena Manzi, who was also a former postdoc and now is a research assistant researcher in my lab, in my group, with my supervision, and also in collaboration with Dr. Elena Knott. And so in this study, we aim at interrogating human serum samples from a patient cohort that included healthy individuals and ccRCC patients from different stages of disease. And we designed a two-model strategy in which we found two different lipid panels that allowed us to detect the disease, differentiate samples between controls and ccRCC patients, and a second lipid panel that allowed differentiating early ccRCC stages from advanced ccRCC stages by means of using support vector machines coupled with the lasso feature selection method. So in this particular study, we pursued and follow more or less the similar QC practices that I have mentioned for the previous studies. In particular, for this study, we consider important the analysis of a chemical standard mix of 14 lipids to evaluate not only the retention time and M over C and fragmentation patterns from different lipid classes, but also to analyze and identify the different ADAC types that were formed based on the mobile phase used in the chromatographic method, which aid in the lipid identification process. As well, in this study, we daily analyze the relative standard deviation for selected features in the intralab QC and intra-study QC samples. And we guaranteed that we had a CV under 20% within every batch and the accumulated batches. We daily perform PCA analysis, and again, we curated the data manually at the end. And the data has also been deposited to the Metabolomics public repository. So these are the three studies and the practices that we have conducted using the UHPLC MS platform. Before getting into TidyMS, I wanted to share with you a completely different strategy that we have implemented in the lab. That is an analytical method that was focused on a marine and atmospheric chemistry application that involved the analysis of seawater samples that were collected in a field campaign at the Cape Verde Island in October 2017 in the frame of an international project. And so in the lab, we developed an ambient mass spectrometry-based metabolomics and target workflow or method by using transmission mode direct analysis in real-time quadruple time-of-flight mass spectrometry. So we gave the name of C-omics to this and target workflow that aim at differentiating the organic composition of dissolved organic matter that was present in samples collected from the sea surface microlayer and the underlying water. So we analyzed of comparing the relative levels of certain compounds, of discriminant compounds. We were able to differentiate the composition based on the composition, the different seawater samples without the need of desalinization. So this study was mainly led by Nicolás and also in collaboration with Malena. And the practices that we use for this particular strategy also involve the analysis of the system suitability blank, process blank, a system suitability sample, the pooled intra-study QC samples that were, we analyzed pools of equal volume molecules of all study samples before and after metabolite extraction. In this particular case, we use commercial seawater as an interlab QC sample. And we had to carefully design the experiment for depositing the samples in the mesh, in the stainless steel mesh that is placed on a rail that is synchronized in order to be able to analyze the samples with the, so that the samples are exposed to the plasma-based source and avoid cross-contamination. We also corrected stigmas with the lowest batch correction, and we applied similar curation filters and steps as I mentioned before. We analyzed the data by means of PCA and TSNE, and the data has also been deposited to the Metabolomics public repository. So with this in mind, at the beginning of 2019, we have conducted already these studies, and in one of the group meetings, we were wondering why don't we try to develop a platform in order to obtain faster and more reproducible results and be able to apply all these data curation steps that we were conducting for each of the studies that we were running in the lab. And that's how, with the goal of pre-processing LC-MS-based and target metabolomics and lipidomics data, retaining only analytically robust enough features for subsequent data analysis and annotation. So all the previous efforts that we have done were the motivation that led us to combine in one free and open tool the quality control practices that we were conducting in our lab, following guidelines proposed by other authors. And once we design the pipeline, we aim at verifying the capabilities of the tool through the analysis of a new suite of candidate plasma reference materials for on-target metabolomics. That is the candidate reference material 8231, currently under development by the National Institute of Standards and Technology, in addition to NIST SRM 1950 metabolites in frozen human plasma. So as I was mentioning at the beginning of 2019, we had a group meeting and we decided that Gabriel Riquelme, who is my PhD student and has also a strong background in computer science, who collaborated at the very beginning of the project with Dr. Pablo Marchi, who at that moment was a student. He's an engineer with also a strong background in computer science. So Gabriel took the lead to design TidyMS in collaboration also with Nicolás, and in collaboration with Dr. Christina Jones from the National Institute of Standards and Technology. And so Gabriel gave the name to this Python-based package for pre-processing LC-MS data for quality control procedures. And this slide, this figure basically summarizes the capabilities of TidyMS, which include the capability of pre-processing raw data by applying feature detection and feature correspondence algorithms, and being able to analyze chromatograms and mass spectra with different tools. As well, the pipeline has, the package has a pipeline for data matrix curation, in which it's possible to perform quality control-based batch corrections, blank corrections, feature and sample filtrations based on thresholds or acceptance ranges determined by the user. And at the end, the pipeline also allows normalizing the data. And along all the steps of the pipeline, the user can calculate metrics such as the RSD, the dip ratio, the detection rate, and the RSD can be estimated also by means of the median absolute deviation. And the user can obtain interactive plots for visual inspections, and these are different tools for quality assessment while conducting and performing the different curation steps. So this scheme briefly summarizes the workflow of TidyMS for data curation, in which the methods for filtering features in the data matrix are encapsulated in the data container, the filters, the correctors, and the pipeline objects. The data container basically stores the data matrix and contains the descriptors associated to the features, such as the M/C and retention time, and the descriptors associated to the samples, such as the class, the order, or the batch. The filter object aims at removing features or samples according to a given metric. For example, the prevalence filter would allow removing features based on a threshold that the user decides in a certain group of samples, for example. The corrector object aims at performing corrections on the data matrix using a subset of samples. So, for example, for the case of the blank corrector, there is a correction that needs to be defined with the sample blank and will be applied to the study samples. And then the pipeline object combines several filters and correctors and applies them simultaneously. And so along all the different steps of the pipeline, the user can generate plots or calculate metrics, and at the end, the curated data can be exported into different formats, such as CSV. And so to illustrate the implementation of TidyMS, I'm going to provide one example data set that was comprised of a system suitability sample and plasma sample classes provided by NIST. So the system suitability sample comprised 5 chemical standards, and the plasma samples included diabetic plasma, a high triglyceride plasma, young African American plasma, and the normal human plasma, which is the SRM 1950. And these samples were analyzed by means of a UHPLC, QTOF-MS-based and target metabolomics method, using a reverse phase chromatographic method, using a UAPLC system fitted with a C18 column and coupled with a water SIBO G2S QTOF mass spectrometer operated in positive ESI mode. So this slide shows the experimental design that was mainly led by Nicolás, in which the study samples, the plasma samples, were split into 6 aliquots. And so basically the sample preparation protocol involved the inclusion of an internal standard, a protein precipitation step by using a mixture of solvents, and then a freeze drying step and a reconstitution in a proper solvent mixture for UHPLC analysis, UHPLC-MS analysis. And this, at the right of the slide, we show the sample list that we designed for analysis, also following guidelines mainly provided by David Brohart and Rick Dunn and collaborators, in which we analyzed a zero injection, a solvent injection followed by a process blank, that was followed by the system suitability QC sample that I mentioned previously. Then we conducted several injections of system conditioning quality control sample in order to condition the LCMS platform, and this number of injections was previously optimized. And following the system conditioning, we analyzed the study samples in randomized and balanced blocks that were injected into a template of pooled QC samples that had a particular design in order to be able to subsequently apply a potential correction due to time-related drift. And at the end of the sample list, we analyze the process blank, a system suitability QC sample, and a solvent to verify the status of the platform and also evaluate potential carryover. And again, the data was deposited to the Metabolomics public database. So the first application that illustrates one of the capabilities of TidyMS is an LCMS system suitability testing. And for those who are not familiar with this term, the system suitability ensures that the analytical performance is fit for the purposes of the experiment. And in this case, as I mentioned before, 5 known chemical standards were used to evaluate the UHPLC-MS platform status before and after the analysis of an analytical batch. So here you see how we previously injected 10 consecutive sample injections of the system suitability sample, which are shown in light blue in the plots, to obtain dispersion values for M/C, retention time, and area of each of the compounds. And so then TidyMS, by means of the raw data preprocessing capability, allows feature detection in the raw data in centroid mode by picking the M/C traces using expected M/C values from the chemical standards, then applies a feature correspondence algorithm and removes features based on the expected retention time of the chemicals from the chemical standards. And then we are able to evaluate the platform status with the samples that were analyzed before in red and after in yellow, running the study samples. And here we see the plots that can be built with TidyMS, and the user then can define which are the accepted amounts or ranges for dispersion of all these parameters. This same procedure was conducted for the pooled quality control samples that were spiked with these chemical standards and also contained the internal standard, and we were also able to check dispersion with this type of plots, interactive tools. The second application that I mentioned at the beginning of TidyMS is the application of a signal drift evaluation and data curation pipeline. So for this case, I'm showing here the different steps that the TidyMS pipeline applied for curating the data that was previously extracted by using Progenesis QI. So we extracted a feature matrix with Progenesis QI, and this is the PCA score plot that it corresponds to the raw data, in which we have included the injection order of these are the pooled QC samples, and these other colors correspond to the other study samples from the candidate reference materials. We see, based on the order of injection in the pooled QC samples, that there is some time-related variance, probably due to signal drift, that can be corrected. And so the first step that we applied in the pipeline was to remove features that would elude at a retention time close to the dead time of the system, in order to avoid including features that would suffer from ionization suppression due to the short elution time. And then we applied the lowest batch correction using the pooled QC samples based on this visual inspection provided by the PCA score plot. We subsequently applied the blank correction and a filter based on the RST in the pooled QC samples. In particular, the RST was estimated with the median absolute deviation. And then we applied a 100% prevalence filter within each class of the study samples, and a subsequent D ratio filter to retain most biologically relevant features. And at the end, we end up with the final matrix composed of 665 features. And after normalizing for total area, this is the final PCA score plot that in which we can see that the pooled QC samples cluster tightly in the center of the score plot. So why we like TidyMS? We like it because it has several tools for interactive data exploration Actually, the previous slide was quite static because I was just showing 2 PCA score plots, but we can obtain metrics and PCA plots after performing each of the curation steps of the pipeline. So it has tools for interactive data exploration, visualization, and analysis using Jupyter notebooks. The curated data also can be easily hyphenated to widely used statistical or machine learning tools such as scikit-learn, which is the one we use for building the PCA models, and we also use it for the support vector machine models that we use in the lipidomics study that I mentioned at the beginning. And it has comparative advantages toward manual pre-processing actions. As well, the source code is fully available at GitHub, and Gabriel would be really happy to receive feedback. We have already received feedback from researchers in different parts of the world, and we are happy for that, about the use of TidyMS. And all the documentation is available online also, so we invite you to check it and try it and provide feedback if possible. And so to conclude with this part of the presentation, we can say that TidyMS provides functionality to perform analysis of raw data of LC-MS system suitability tests and data curation on untarget metabolomics datasets. The pipeline was successfully implemented to pre-process data corresponding to the NIST candidate reference material 8231 and the SRM 1950 for metabolomic studies. It can be customized or fit for purpose according to the specific application. And the package offers a rapid and reproducible workflow that can be used in an automated or semi-automated fashion. And it is an open, free, and locally executed tool available to all users. Besides, it's a tool that is currently being updated, and we are introducing new features, as Nicolás will later describe in his presentation. So with this, I would like to acknowledge all my collaborators in Argentina and overseas, and the funding agencies from Argentina and from Europe, and mainly to the Metabolomics Society, to the EMN network, who provided us with this great opportunity to share what we are doing in this part of the globe. And in particular, I want to give a big thank you to all my team members who trusted me when they joined the group, and I started alone in 2014, and my first student started in 2016. So they accepted the challenge of starting in a new group, in a new institute, in a new lab, and working in a field that was not highly developed in Argentina. And so I want to thank all of them. Gabriel Riquelme and Nicolás Zabalegui, I've already mentioned them in the presentation. Maximilian Rey has just joined the group the 1st of April, and so he will do his PhD on the project of dengue disease. Malena Manzi and Manuela Martinevsky are both assistant researchers of CONICET with my supervision, and they were former postdocs. Malena was one of the first, together with María Elena Not, who joined the group, followed by Nicolás and Gabriel. And I want to also thank Mariela Garcia, who also recently joined the group last year during the pandemic. And we have only met also virtually so far. And a big thank I want to give to Mariela Videla. Dr. Videla helps me to run the mass spec facility at the center, since we also provide services to the local industry and regional industry and to the academia. Unfortunately, there are very limited number of instruments of high resolution mass spectrometers in the country, and so we play also an important role in providing services to the academia. And I want to thank my former postdocs. And so then, before finishing the presentation, as I mentioned in the outline of the talk, I will share some news about the Latin American Metabolic Profiling Society and thoughts about how MS-based technologies can be transformative for disease diagnosis and contribute to global health equity, not only MS-based, but also NMR-based technologies. But unfortunately, technological developments utilizing omics-based technologies have not been shared or distributed equally. And these are some thoughts that we share with the Latin American Metabolic Profiling Society members, who is a young network that pursues strengthening collaborations across the Latin American region, as well as promoting emerging technologies and practices that help to make science more reproducible and open. We invite all of you to check our website and to follow us on Twitter, since in the near future we plan to provide information to expand our network and have more members from Latin America. So far, the LAMPS members initiatives that have been conducted since 2014 were different meetings with workshops that were held in Peru in 2014, in Argentina in 2016, and in Brazil in 2018. We were going to have our 4th meeting to be held in Colombia in October 2020, that was and has been postponed due to the pandemic. And we have coordinated so far 3 hands-on metabolomics courses at Cirion CONICET in my lab. We have received international professors from abroad. For example, we have received Arthur Edison and Facundo Fernandez joining us in the teaching courses. And we have also held different courses in Argentina, Brazil, and Colombia, having students from Brazil, Argentina, Colombia, Paraguay, and Uruguay. So we invite you to check the website again to read more about the challenges that we have to face to move forward the metabolomics field in Latin America. The first challenge is probably common all over the world, which is to guarantee accessibility and equality to all scientists. But then there are several and particular challenges that are related to the Latin American region, which includes the instrument choice, since the Latin American market is not very attractive for vendors, and there are not local representatives of the vendors in all over the countries. Then we have to face challenges regarding accommodating variability in physical and financial climates. We have sometimes limited access to materials and supplies, or we have to face high prices and delays in receiving reagents and supplies. We also face challenges regarding the access to open source software and data repositories, the challenge associated to corporate recognitions of the benefits of supporting resource-limited laboratories and institutions, and the transfer of technology and expertise. I invite you to go and check our website and read more about this if you're interested in. And so who we are, these are the LAMPS founding members. In particular, I want to acknowledge, and as we all founding members want to acknowledge Elaine Holmes, who supported us from the very beginning and participated in all the LAMPS meetings. And then we have Julien Wist from Universidad del Valle in Colombia, who runs an NMR facility and research group, and also Mónica Cala from Universidad de los Andes in Colombia, who is the head of Medcore, a mass spec facility that has recently been started in Bogotá. Then we have Paula Burdizo and Alejandro Vila from Rosario in Argentina. Alejandro actually organized the LAMPS meeting that took place in Rosario in 2016. We also have Pablo Eichenberg, who is my colleague at Cirion CONICET, leading the mass spec, the NMR-based metabolomics group. We also have Martín Arán from Phil CONICET in Argentina, and Guillermo Moina from Universidad de la República in Uruguay, and Ian Castro Gamboa, who is from Brazil. And so before I finish, I wanted to share the good news, that is that the LAMPS has recently become a new international affiliate of the Metabolomics Society, and we hope that this affiliation will enable global collaborative and training opportunities and provide greater representation of Latin American members in the society. And with this, I want to finish with a big thank you for your attention. Muchas gracias. This is my email, and this is the website of our institute. And I leave now the space for Nico to continue discussing strategies, quality control strategies with TidyMS.
Okay, thank you very much, Maria, for the very good talk. The type of QC practices that you do makes me question what I'm doing. There's a lot of things that you do when it comes to QC, but I think in those lists, I could only take 3 that I do. So it's always a learning curve for each and every one. So without wasting any time, let's move to our next speaker, who is Nicolás. And just a brief bio about Nicolás. Nicolás is currently a member of the biological mass spectrometry group led by Dr. María Incibión and Konek. In 2017, he was graduated from the chemical science from the University of Buenos Aires in Argentina. And since then, he is a PhD student in the Inorganic, Analytical, and Physical Chemistry department at the university. His research work is focused in the development of mass spectrometry-based analytical method for the different application in untargeted metabolomics. In particular, he has conducted untargeted metabolomics profiling, footprinting studies focusing on early diagnosis of clear cell renal cell carcinoma using ultra-high performance liquid chromatography coupled to quadruple time of flight mass spectrometry. He has also gained experience in developing analytical methods using ambient ionization techniques, such as direct analysis in real time, following rigorous quality control practices. During his training as a PhD, he has analyzed different types of biological samples, including from in vivo, in vitro models and human serum sample, as well as samples from ocean environment. He is the member of the Metabolomics Society and is a co-author of 6 peer-reviewed publications and has presented his research work in different national and international conferences. I would like to say that if you have questions for Maria and also questions for Nicolás, please post the question in the chat box and we'll attend to them at the end. And in this webinar, Nicolás will present how liquid chromatography mass spectrometry-based untargeted metabolomic studies involving biological samples may lead to detection of tens of thousands of potential metabolic features, such as retention time, mass-to-charge ratio pairs at an initial stage of the workflow. However, data needs to be pre-processed in a reproducible way to remove biologically non-relevant features and thereafter obtain cleaned matrices suitable for the subsequent statistical analysis. And with due fact, it's over to you, Nicolás. And thank you.
Thank you, Cici, for your kind presentation. Hello, everybody. Thanks for joining. In this short talk titled Improving Data Quality in Metabolomics Workflows, I will present the implementation of TidyMS on a much larger cohort corresponding to patients with renal cell carcinoma and healthy individuals. I will also introduce some additional practices that are now available in TidyMS. First of all, I would like to give you a brief outline of this talk. Taking advantage of Eugenia's previous presentation, I will first highlight some aspects of kidney cancer disease, focusing on renal cell carcinoma. Regarding our untargeted metabolomics study, I will provide some information about sample collection and storage, and after that, I will describe the patient cohort. Then I will continue explaining the experimental design together with data preprocessing. At the end of this section, I will show some additional QC practices we apply to account for sample stability over time and to evaluate the response of features towards dilution of pooled quality control samples. I will end the talk with some conclusions and stating what we plan to do in the near future. So, kidney cancer is a disease of dysregulated cellular metabolism and is the 15th cause of cancer-related death worldwide. As you will appreciate from the bar plot of 2020 incidence data worldwide, men are more affected than women. In this plot, the gender ratio is shown for each of the 5 continents to highlight this fact. As María Eugenia previously mentioned, approximately 90% of renal tumors are renal cell carcinoma. So renal cell carcinoma is comprised of several histological subtypes, being clear cell renal cell carcinoma the one with highest incidence, and the one which accounts for most cancer-related deaths. Other important subtypes include papillary RCC and chromophobe RCC. The staging of renal cell carcinoma reflects the tumor size, the extent of invasion outside the kidney, the involvement of lymph nodes, and whether the tumor has metastasized. So it's possible to classify these 4 stages in early and late stages. And in the plot on the right, the percentage of people who are alive after 5 years from being diagnosed is represented for each of these stages. I would like to recall that the disease is potentially curable by surgery, but only at early stages, as surgery is typically reserved for localized disease. So this is the untargeted metabolomics study timeline, and now I will provide some information of each of these stages. Regarding serum sample collection, serum samples were collected by the Argentinian Public Oncologic Serum Bank, which collected samples from 2 institutions, Hospital Italiano de Buenos Aires and Instituto de Oncología Ángel Rojo. Serum samples were collected following the guidelines approved by IRB and after signature of the corresponding informed consent. Regarding the cohort description, in this slide, you can see a table describing each of the sample classes employed in this study. Also, age ranges and median values are shown. Clear cell RCC samples were collected from patients covering all 4 stages, and in some cases, a second sample was collected after tumor removal. Other histology samples were utilized, such as papillary RCC and chromophobe RCC. And other less frequent histologies look here as other. The pie plot on the left represents the disease incidence by gender in Argentina, and the plot in the middle represents the gender distribution in this particular cohort. Samples employed were age-matched, as you can appreciate from the violin plot on the right side of the slide, which also shows the age distribution by gender. As regards the untargeted metabolomics method, the sample processing steps involve protein precipitation, centrifugation, separation and freezing of supernatant at -80 degrees, and sample reconstitution after supernatant lyophilization. Processed plants were also processed in parallel, employing the reconstitution solvent, and pooled quality control samples were prepared using all the samples involved in this study. Samples were analyzed with a water SIBO G2S Q-TOF mass spectrometer, operated both in negative and positive ion mode in the M/C range between 50 and 1200. In this presentation, I will focus only in negative ion mode data. The reverse phase chromatographic method applied is the same method we used in the metabolic footprinting study that María Eugenia described earlier, in which we analyzed samples from in vitro models and from human serum. Regarding the experimental design, the study comprised the analysis of 258 samples. 26 samples were analyzed on 10 different batches, and each batch corresponds to a different experimental day. Samples were randomly distributed across all batches within a class balance design. This sample list is similar to the one which was employed when analyzing the NIST plasma samples described by María Eugenia. The system suitability quality control sample followed by a process blank sample were injected before conditioning the analytical platform with system conditioning quality control sample injections. Intrastudy quality control samples were injected, in this case, every 5 or 6 samples, so that time-related effects could be corrected by the lowest method. The following slide, this slide summarizes all the preprocessing steps applied to obtain features potentially identifiable and biologically relevant. At the beginning, a feature matrix containing almost 8,800 features was obtained by means of Progenesis QI software. After that, A retention time filter was applied to remove features with very low retention time, which usually suffer from high ion suppression effects. The process goes on with the lowest batch correction, where only features with lowest corrected area values can be preserved. After subtracting the blank, a coefficient of variation filter was applied, discarding all features with more than 20% of relative standard deviation in intrastudy QC samples. After that, a prevalence filter and a D-ratio filter were applied. The D-ratio filter is aimed at removing features with low biological relevance. The three last steps in light blue were recently added to our current pipeline and were aimed at obtaining potentially identifiable features. So finally, a feature matrix of around 1100 features was obtained. To illustrate why we conducted different steps in the pipeline, here I'm showing a PCA score plot of intrastudy QC samples before and after data preprocessing. Clusters of different batches can be visualized in the plot on the left, whereas after performing the batch correction, these clusters disappear and random error is the only source of variation. In the following slides, I want to point out that in TidyMS, we can easily change the sample label to evaluate potential confounding factors. In this slide, the PCA score plot was made employing class membership as label, but we can do the same with batch order, for example, or gender, or any other metadata information we have, and check that no trends or clusters are observed in the plots. As you can see, all intrastudy QC samples lie in the center of the plot after performing this data preprocessing step. So after acquiring this data at the end of 2017, I started preprocessing all the data in 2018. However, when we started analyzing this data, we decided to hold on with data preprocessing and wait until TidyMS was fully developed, as we didn't want to perform manual curation of data this time. I was about to conduct additional experiments which were planned for the beginning of 2020, but unfortunately these experiments were delayed almost 1 year due to the pandemic. The first of the additional QC practices we wanted to implement was evaluating sample stability over time. We were interested in reanalyzing sample extracts from 2017, which were stored at -80 degrees, to see which features could be detected again, which proportion of features. As a second goal, we also were interested in those metabolites that exhibit monotonic response towards full QC dilution. So that's why we decided to perform intrastudy QC dilution experiments. Regarding sample stability evaluation, we analyzed again sample extracts which were frozen at -80 degrees, and after extracting a new feature matrix with Progenesis QI, we decided to search each one of the almost 1100 features in this new feature matrix. We did this search considering a retention time tolerance and an exact max tolerance, and we also employed causing similarity in the case of isotopic pattern matching, and added the convolution information when doing the final manual revision of the feature matching process between both matrices. It's important to mention that almost 60% of features could be detected again in this experiment. Then we decided to refine this search by choosing only those features that exhibit monotonic response towards pooled QC dilution. So now I will show you briefly how this experiment was done. Pooled QC sample extracts stored at -80 were serially diluted 4 times. Each QC dilution sample was injected 3 times, and in each case, Pearson and Spearman correlation coefficients were obtained. I want to say that now this option is available in TidyMS. After that, features with Spearman correlation coefficient higher than 0.98 were retained. So of the 685 detected features, 488 features showed a Spearman correlation coefficient higher than 0.98, exhibiting thus a monotonic response towards pooled QC dilution. Then by means of visual inspection of the regression curves, we could identify a subgroup of features with a clear linear response towards pooled QC dilution. We plan to check normal distribution of regressions residuals by implementing this practice in TidyMS soon. To conclude, the pipeline was focused on retaining analytically robust features that may be potentially identifiable. First, preprocessing pipeline allowed retaining almost 1,100 features, metabolic features containing potentially relevant biological information. The package allowed visual inspection of data, as well as feature or sample elimination in a versatile manner, because the user can go back and forward easily when doing all the preprocessing steps. So after the stability-related filter, 685 metabolic features were retained. And then after the correlation filter, only metabolic features that exhibited higher correlation coefficients with serial dilution of intrastudy QC samples were retained. In the near future, we would like to preprocess positive ESI mode data and integrate all this data with negative ion mode data. And we also plan to apply multivariate statistical models to find reduced feature panels capable of sample classification. We would like to identify the discriminant feature of these panels and this work with data analysis and biological interpretation of results. So I would like to thanks to all my colleagues from the Monge team and to my supervisor, María Eugenia, to all the funding agencies, to Dr. Puricelli, Dr. Jimenez, Dr. Knott, and to the Public Oncologic Serum Bank, and especially to the Early Career Members Network of the Metabolomics Society for giving me the great opportunity to show my work here, our work here. So thank you very much.
Thank you, Nicolás. Let me also thank Dr. Monge for her great lecture, and thank you, Nicolás, for your talk as well. So let's move on to the first question. So the first question is for Dr. Monge. Many congrats on this amazing lecture. Is there any plan to make the TidyMS package available for the art software?
For the art software?
Yes.
So far, the package has been developed in Python, and it's open, and I show already, and in the publication, you can find all the links also to the GitHub and the documentation. Everything is available. So so far, no, we are moving forward with Python. I mean, all the group has been trained in using Python. And not in the short term, I would say, but maybe why not? I mean, it's not impossible.
Great. The second question is again for Dr. Monge. A great presentation. I was wondering if the TidyMS will also be developed for the GCMS data.
That would be great. Unfortunately, we don't have a GCMS instrument in the center. So, because ideally we would like to try and see how the tool performs with our data. But I mean, we can. I mean, if desired, we can think about expanding. And also, Gabriel, when he started with this development, he also started by designing a pipeline just for DirectMS analysis, and then we were like more ambitious and said let's focus on LCMS, which is the most prevalent strategy that we use in the lab. But yes, I mean, it can be expanded to other platforms.
Perfect. Another question for Maria. Having in mind that there is no standardization yet for the use of systems suitability samples, I would like to know from your side, what are the criteria that you employ in your lab to decide if the system is okay to continue the analysis on one specific day? Meaning, do you consider the CV on internal standards? Do you extract also unknown features? Do you consider retention time shift, etc.?
Okay, yeah. Yeah, actually, this is like topic in which there is still no consensus. So far, in the case that I have described, that in which we exemplify the capabilities of TidyMS with the system suitability sample, we use twofold the standard deviation of the mean for the as an acceptance criteria for dispersion in retention time, in M/C and peak area, and 3 seconds in retention time. But this is something that has to be probably defined according to the instrument that you have in the lab and how it performs in terms of reproducibility. So this is what we have used so far. Regarding system suitability, in a current study that we are conducting now, actually Gabriel is running samples now in the lab probably, or maybe he's attending the seminar. We noticed that, for example, in some cases, it was better to use pooled QC samples that were spiked with chemical standards for avoiding using too much time in conditioning, in analyzing the system suitability samples that are just chemical compounds in solvent, and then stabilizing the LCMS, conditioning the LCMS platform, for then subsequently analyze the study samples in a template of pooled QC samples. So we found out that another strategy that we may apply based on the study, on the case of study, is to evaluate the dispersion of an internal standard or a group of internal standards, and also these known chemical standards that are spiked into the pooled QC samples, instead of analyzing them just in solvent. So that's another strategy that we are evaluating right now.
Great, thank you, Maria. Another one on TidyMS. Is it correct that TidyMS is not estimating the proportion of peaks missed by non-targeted pre-processing? I'm asking because we recently published on our package doing that, and it seems like it will be nicely to complement your TidyMS package.
So
and our package that they publish is called MZRAP.
MZRAP. That would be great. So you mentioned that so there are if it captures features that are not.
Yes, sure. Is it correct that TidyMS is not estimating the proportion of peaks missed by non-targeted pre-processing?
Proportion of peaks that are missed by the pre-processing.
Yeah, by non-targeted pre-processing.
It doesn't have that capability, but I may need to. I would like to read. I will definitely read this new publication on this other strategy to understand better the question, because maybe we can complement and we can include that capability in TidyMS. I mean, so far, TidyMS, let's say the pre-processing, the curation pipeline that is applied on the data matrix that is already extracted with another more solid software such as MC-Mine. We use Progenesis QI in these cases, but MC-Mine or XTMS, but it still doesn't have the capability of extracting all data matrix from an untarget study. I mean, so far, the pre-processing of raw data applies to known chemical standards that are used in a system suitability QC sample. And we aim at checking and verifying the feature correspondence algorithm that we use and test it in a larger number, in an untarget workflow, let's say, to be able to see if we can extract all the features as MC-Mine would do or XCMS would do in our locally executed pipeline.
Great, thank you. So the next one is, I'm curious about the prevalence filter that you said to 100%. If that is across all samples, don't you risk eliminating the differences between groups?
I mentioned that the 100% prevalence filter, it was not written, but it's an intra-class filter. So it's within each class of the study samples.
And then there's
in the case of in the example that we provided, they were just 4 samples that in order to have more variability, that's why Nicolás split each of the 4 samples into 6 aliquots. And since they were just the same sample, that's why we applied that type of filter for that particular application. But this filter can be changed based on the study objective. I don't know.
There's a second part to the question. They're asking why use an RSD filter if you could also use the D ratio? Isn't the point of the D ratio to avoid setting a hard threshold?
The D ratio is used at the end in order to retain those features that may have more maybe biologically relevant, so that we retain, we want to exclude features that have larger variance, like technical variance, than the variance provided by the study samples, like the biological variance. So I don't know if I answered the question.
Great. The next one is
RSD in the pooled QC sample. The RSD is a filter in. Sorry, Evelina. The RSD
go ahead.
Applied. It's a filter for the pooled QC sample, so it considers just the technical variance.
Have you tried to do the statistics without removing features? Is the result any different?
Well, we actually, at the very beginning, when we started with the first study, and we were practicing, and even like the different group members were learning because they didn't have knowledge on metabolomics, and we were trying to check what would happen if we just use raw data and do statistical analysis on the raw data, and we end up finding that you can find as potential biomarkers noise or artifacts, because using a full matrix of raw data, there are many signals that introduce noise and are in detriment of the feature selection process for finding potential biomarkers. So what we want is like to work with the more robust as possible feature matrix, so that we end up with robust and analytically robust features that may be potentially annotated or identified with the highest confidence level as possible.
Great, thank you. The next one is for Nicola. Regarding the QC dilution, do you have any kind of control for low intensity features? Because linearity would work fine for low features that are still detectable after all dilution steps, but not in the case of low intensity features.
Thank you for your question. I believe that those low intensity features are removed in the first stages of our pipeline. So, however, at the end of the whole process, I checked every regression line visually, and I think we are not having problems with low intensity features because I checked those values. But
thank you, Nicola. There's another one for you.
Yes.
What are you looking for when injecting conditioning QCs? What are the conditions to be met?
Oh, thank you. We have seen that when we start doing the experiments, the analytical platform is not well stabilized, so we need to condition for example, the column with samples similar to the samples we are working with later. So in fact, when we look at the PCA score plot, we can see how the quality control samples and the intrastatic, how this drift is taking place. And so I'm not sure if I'm answering the question properly, but
may I add something?
Yes, please.
Yeah, I want to just briefly add that we condition not just the chromatographic system, but the LC-MS system as a whole. And what we use is just visual inspection of a PCA score plot, and we see how pooled QC samples start to cluster once the system has been conditioned. And that's how we can easily define the minimum number of injections that we need to do of pooled QC samples for conditioning the platform.
Perfect, thank you. So the next one is for both of you. May I know if TidyMS is sensitive to batch design, for example, having a specific number of QCs, presence of pooled QCs, pooled batch QCs, etc. What if pooled batch QCs are not present in the acquired data?
Nico, do you want to?
Yes, of course. If those conditions are not met, I mean, only features with certain conditions will remain after the pipeline. So you necessarily need to employ one quality control sample after conditioning the platform, and you need to bracket. You need at least 4, if I'm not wrong, 4 quality control samples to perform the lowest correction.
Thank you, Nicolás. This one is for Dr. Monge. How do you treat the blank curation of the signal? It is clear from the pooled samples, but a curation by blank sample is a big challenge for me.
Well, that's also a criteria that is not unified in the community, and we have conducted different strategies, but mainly we use the process blank and the solvent blanks, and we see if there is a signal that is, for example, one strategy would be if there is a signal that is present in the blanks, in the blank samples, and it's also present in the study samples. But if the intensity is more than 10 times in the study sample than in the blank sample, then we would subtract the peak area. Otherwise, we would set that feature to 0. So, but this is a threshold that can be changed. It can be 10 times, 5 times, also depending on how is your system. And we typically try to be to remove most signals present in the blanks.
Great, thank you. Another one for Dr. Monge. How do you deal with electronical noise? How do you curate it?
I would say that electronical noise is always, and we live with that, and it's not included as a. We don't consider it in this pipeline because we assume that the instrumental, the technical variance by, for example, checking how the pool QC samples respond in time, in which we may see that the signal may decrease due to a sensitivity issue or may increase due to a carryover issue. And those are usually changes or fluctuations that are much larger than the instrument noise. Fortunately, when we turn on, when we put the instrument into operate at the beginning of the day, and we calibrate the time of flight, and we check the signal of the log spray, that we use to correct the log mass, the compound that we use to log the mass during acquisition in the LC-MS system. At that moment, we already know if there is like a big issue with the instrument in terms of noise. But otherwise, the other fluctuation or variation is much more important.
Thank you, Maria. We've got one last question. How do you normalize the data? Can you normalize between data sets that are obtained on different dates?
We end up at the end, we have been normalizing by total area at the end of the pipeline, but normalization is itself an issue that may also influence on results. And there are very nice review articles from the groups of Coral Barbas and Bisma, I don't know how to pronounce his name, but that deal with this issue. And so far, I don't know, Nico, you were going to say something?
No, I just want to add that the batch corrector includes a normalization between different days.
Yeah.
Perfect. Let me just thank you one more time. Thank you so much for your great talks. We really enjoyed both of your talks. And let me also thank everyone who joined the talk for today. And also, guys, keep posted for the upcoming webinar in May. More information will become available on our platform. Thank you so much once again. Hopefully we will see each other soon in upcoming conferences. Hopefully.
Yes.
Bye, everyone.
Well, thank you very much. Thank you very much for this opportunity and invitation. And thank you very much to all of them who stayed up to the end.
Perfect.